That, and now I'll try to share the screen.
Yeah.
I can do these, like, these are great. What does this mean? We never talked about that.
Because she's
Rude.
nutrition
And then this one.
in informatics. Was something that needed a.
Is it sharing on that? So stop sharing video.
Oh, okay.
I
Yeah,
did
it's
this.
which is a dietitian.
Somehow I got this weird
Okay, cool. Well, we got going. Okay.
And it'll pick me up sound wise.
It should. I'm actually hoping, you're the first lecture we're recording because
Okay.
you're the first new person this year and I have all the ones we recorded last year, so.
Cool. Okay.
And hopefully it's recording something.
So I'm ready to go?
You should be good to go, yeah.
All right. Sorry for the slow start.
Nah, you're good. I mean, it's all good. I'll be right over here.
Okay. So pleasure to be here. Excited to share some of the things that I'm interested in, and hopefully I can convince you that you might be a little interested in it. So Dave Arnold. So I'm been here just a little over a year. So I'm a physician scientist. So I'm probably going to be a little bit more clinical than a lot of people have been on in other lectures. I joined as a physician scientist, moved my lab here, and also and the executive director of NextGen. You've probably seen the big shiny building. I also help co-direct the PhD program. So some things that I hope to cover today, and I hope you will be willing to stop me if you have questions. It's really, there's no such thing as a bad question unless you don't ask it. Some of the things I hope to talk about today is it's not showing you. You know how I changed that? Because it's not tracking with me.
Oh, really?
Yeah, that's weird, right?
Let's see, what do we do you whole screen? Oh, screen 2.
Yeah, maybe that's.
Here, let me pull up there.
Okay. See it go forward. That's better. Okay, perfect. Okay, yeah, okay. So, some things that I hope to talk about today, and hopefully this is not redundant with what you've already heard, but if it's a little redundant, that's okay. So I'm very interested in what I call translational neuromuscular physiology. So we're going to do just a little bit of review of normal neuromuscular physiology. And in regards to what my favorite part of anatomy is, the motor neuron and the motor unit, what that is, how the brain modulates contractility and force production of muscle. And then one aspect I'm becoming more and more interested in is neuromuscular junction transmission. And then as a clinician, a physician scientist, I still do clinical work. So I take care of patients with neuromuscular diseases. And so what is that? So that's disorders that affect the peripheral nervous system. So things like motor neuron diseases, peripheral nerve diseases from neuropathies, neuromuscular junction diseases, things like muscular dystrophies. And so I'm going to talk about 2, maybe 3 cases, depending on how fast we go, of some interesting kind of scenarios that I've worked on research-wise, but also as a physician. So a high-level overview of the neuromuscular system. So we broadly divide the nervous system into central and peripheral. That everybody in here probably knows at this point. So the neuromuscular system is basically anything from the spine peripherally. So the motor neuron peripherally. I'm going to be very motor-centric today in this talk. So most of the things that I work on affect muscle function, motor strength, coordination, balance, things like that. Not really going to focus on things like autonomic function or sensory, although clinically I do that. But in these representative cases, I'm not going to talk too much about that. So the main focus point is the motor unit. So that is the smallest functional unit of the peripheral nervous system. So what is a motor unit? So basically it is a group of muscle fibers. So if you were to look at my thumb muscle, so the abductor pollicis brevis, that's the muscle that lifts my thumb up like this. So when I want to do like opposition for grasp. So that muscle is supplied by roughly 200 to 300 motor neurons in my cervical spinal cord. They send those projections, axons out to the muscle and connect by a number of neuromuscular junctions. So a typical motor unit, the number of muscle fibers that it connects to varies by muscle. So in extraocular muscles, it may be as few as 6. In a large like quadricep thigh muscle, it may be thousands per single motor neuron. Motor neuron, as I mentioned, is my favorite cell in the body. It's a really amazing cell. If you think about the size of the cell and the length of the cell, the length is up to 10,000 times the size of the cell body. And that's a pretty amazing feat because this is a cell that you're born with the number of motor neurons that you have for your whole life. So it's post-mitotic. There's no known way to replace it. And it has to connect to all these muscle fibers. And also, when you're contracting, it will actively like depolarize action potentials up to 100 times a second to optimally activate your muscle. And so it is a very energetically active cell, and it has to do that job your whole life. So how is muscle force modulated? So there's really two main mechanisms that the central nervous system via the motor neuron modulates muscle force. So I mentioned 200 to 300 motor neurons in this muscle of my thumb, the abductor pollicis brevis. So that's one of 600 muscles in our body. But so as I slightly contract and then go to a stronger contraction, my nervous system will activate additional motor neurons. And each of those motor neurons each has its own motor unit, so groups of muscle fibers. Those muscle fibers that are connected to that motor neuron only connect to a single nerve cell. But each nerve cell connects to 6 to thousands of muscle fibers depending on the muscle. So as I increase the rate of contraction, the strength of contraction, the number of motor units, motor neurons become activated. And it typically is in a very orderly fashion. So small motor neurons to large motor neurons. Different types of neurons have different types of characteristics. Some can fire very fast, some don't fire very fast, but then they can resist fatigue. And the muscle fibers that are connected to those motor neurons have similar properties. So you probably have heard of like glycolytic processes and oxidative. Don't get down those weeds, but basically, depending on the task that muscle is trying to do, you use different substrates of, whether it's fats or it's glucose for maintaining the energy balance of the cell. And so different motor units have different, basically, superpowers, whether they can fire fast or whether they can fire for a long time. And some are kind of in the middle where they can do a little bit of everything. And so modulation of force is by the number of motor units, but also the firing rate. And so this is something that we are in particular very interested in, is how rate coding or firing rate modulation affects muscle contraction. And so the faster you fire, the more force you can get from the muscle. And so I have this figure here. You're probably wondering what the heck I'm trying to show there. So this is basically a readout where we are connected to a foot of a mouse, and the mouse is anesthetized, and we can stimulate the nerve and the muscle contracts. If we stimulate one time, you get this small red blip here. So have people heard of twitch contractions? Well, a twitch contraction is a single stimulation from the motor neuron activating its group of muscle fibers. If you stimulate that motor neuron, or if the motor neuron activates on its own when I contract my muscle repetitively, you can see as we stimulate more frequently or at higher hertz, going from 5 hertz to 30 hertz to 45 hertz, you can see how the twitches are starting to stack up on top of each other. So basically those twitches summate, and then the force is modulated. So the faster you activate the neuron, or the faster the neuron fires on its own during voluntary contraction, the more force you get. And so the number of motor units, each muscle is controlled by a single neuron. So if that neuron doesn't fire, that muscle isn't activated. And then if that muscle is activated, the faster you activate, the more you get from it. So those two parameters are modulated by basically descending input from the nervous system. And those can be disrupted in various neuromuscular diseases, in which I'll show you some. And really, this is our main interest in the lab, and I'm not going to talk about that today actually at all, is how the motor neuron fails across the lifespan. So this is a cell we can't replace. We know that there are losses of motor neurons, and that the motor neurons lose their ability to fire in a way that optimizes muscle function. That's things that we study in the lab, but not going to show that today. So hopefully that's clear. Any questions about that? Okay, so we can measure motor units in patients, in animal models, in companion animals, and this can be used to look at how well the neuromuscular system is working. So this is a electromyography recording. So basically this is a recording electrode while the patient is contracting their muscle. So this is a anonymized recording, and you couldn't tell who this was because they all kind of look the same across people. But this is a patient with no neuromuscular disease that we can pick up or detect, and we see a normal motor unit. So keeping in mind, each muscle is controlled by a single neuron. Each motor neuron controls groups of muscle fibers. So when those motor neurons depolarize, we see what's called a motor unit action potential. Has anybody ever heard of motor unit action potential? People talked about that yet? Probably. EMG is probably a thing that a lot of people have talked about. But this is from a patient, and normally the sound works, and so it's a little bit more interesting to watch. But you can see groups of responses here, kind of firing. I'm gonna play that one more time. I don't know if there's any way to do sound.
I think when you share the screen, you need to check that box that says.
Yeah, let's do that because I think that would be kind of nice.
Yeah, you should. So end the share, and then we'll try it again. And then share sound.
Yeah, perfect.
And then I think we yeah.
We need that?
We'll click it, see what
I'll
happens,
do
I
it.
guess.
Sounds good. Okay, let's see if this works here. Okay. So you can hear they have audible sound. In clinic, the sound kind of can tell us what's going on, whether it's a nerve problem or a muscle problems. And so these groups of squiggly lines are what we call motor unit action potentials. You can measure those in a lot of different ways. We look at the number of the activation, how many different shapes of motor units there are, how fast they fire, and in different disease contexts, that firing rate and recruitment can be altered. Also, the size matters. So with losses of motor units or motor neurons, muscle fibers get disconnected from the nervous system. The motor neuron is the final link between the nervous system and muscle. And through collateral sprouting, the remaining motor neurons and motor units can expand their territory, compensate for those losses, and reinnervate muscle fibers. And so they can get bigger if there's nerve loss with chronic kind of repair, collateral reinnervation. In opposite of that, if you have a muscle disorder where muscle fibers are dying or undergoing necrosis, the size of the motor unit will get smaller. And we use those kind of patterns to diagnose if people, like, do they have a neuropathic problem or a myopathic problem? And so this is something that has been around for decades, but still very, very relevant in clinical work. And we also do this in research as well. Let me share my screen. So I want to tell you a little bit about my pathway. So I kind of decided on medicine when I was in high school, kind of got on that path. Didn't really know what that meant, but a family friend suggested I do that. So I went to pre-med and I went to med school and I decided on a specialty called physical medicine rehabilitation. It's focused on function, really across different systems. And so it has some overlap with neurology, has some overlap with orthopedics and sports medicine. I actually thought I would be a sports medicine specialist. But then during my residency, so that's the training after med school, I fell in love with the nervous system and neurological disorders. And so I went to Ohio State to do a nerve and muscle disease fellowship. So that's subspecialty training in disorders of the peripheral nervous system, and was there for a little over 14 years and then just joined just recently. But my path to research was very odd. So growing up, I didn't have any exposure to research and some of the things that you're seeing now, because I went to a very small school, University of the Cumberlands. And so I really wasn't exposed to research until I was faculty at OSU doing clinical work. I was a clinician, full-time focused on taking care of patients, saw people doing very cool research, and then decided to kind of make a little bit of a pivot or a major pivot to research. And over time, developed my own lab program, and that's kind of what led me to be here to be part of NextGen. So what is neuromuscular medicine? So as you can expect, it is, so it's showing the weird, is it okay if it's just showing the wrong view? I'm sorry. Is that okay?
I mean,
Can you see it okay?
you
I'll
have to
just go.
see it.
Okay, so you can anticipate what I'm going to say, so that's good. So as you can expect, neuromuscular medicine is a field of medicine, usually under the umbrella of neurology or physical medicine rehabilitation. Most people do the neurology path, so I did physical medicine, which was a little atypical back then. But basically take care of any disorder of the peripheral nervous system. So this can be neurodegenerative, this can be traumatic, this can be genetic, it can be autoimmune, or a mixture of all of those. And so anything from the spine out to the peripheral nervous system, sensory, motor, autonomic, all of those are kind of captured in neuromuscular medicine. So it's kind of a niche space on mostly rare disorders that maybe some of those you've heard of. So some I want to mention, In passing, people have probably heard of Lou Gehrig's disease. So that's a neurodegenerative disease that usually is age-related, and it affects the motor neurons of both the central nervous system and the motor cortex, as well as the spinal cord. So people present with very severe muscle atrophy and weakness, slowness of movements, spasticity, and then there's diseases of the peripheral nerve, Charcot-Marie-Tooth disease. So what is Charcot-Marie-Tooth? It's 3 last names of the people that described it in England and France. But basically, it's neuropathy related to hereditary, inherited gene variants that make you more susceptible to degeneration of the peripheral nerve. And then spinal muscular atrophy is actually the disease that I grew up in that space as a scientist. So I was part of a team that developed a therapy that, if we have time, I might talk about a little bit, but this is the most common genetic form of death in infants, but it's a very rare disorder. About 10,000 people in the US. Generally affects infants, but actually has a range of, probably know the term phenotype or phenotypic severity. So the clinical presentation of a disease is what we call phenotype. So it has a range of severity in regards to phenotype. So that's kind of the range of things that some of the things that I deal with. Currently, my main areas of focus are ALS or Lou Gehrig's disease is one of the main focuses, but I really do all of neuromuscular here as a clinician, which is really a relatively small part of my job currently. So case presentation. So this is a case that I saw earlier in my faculty position, and I thought it would be a nice thing to maybe talk about today. So this woman presents really with lifelong problems of intermittent weakness, but between bouts of weakness, she also had some persistent weakness as well, but it was very come and go or paroxysmal, episodic, and no clear family history. So healthy parents, healthy children, and her siblings didn't have any obvious weakness. Examinations that were striking, some of these words you may not recognize, I'll clarify what they mean. So this picture, what we're looking at is her face at rest and her face trying to smile as big as she can. And so you can see very kind of drooping of the eyelids. That's what we call ptosis. It's not ptosis. A lot of people say it that way. It's ptosis. But drooping eyelids is equally accurate description. Ophthalmoplegia. Does anybody know what that is? Ophthalmo means eye, plegia means weakness. So basically, it's the muscles that move our eyes when we're tracking objects. And she had very severe ophthalmoplegia, which I'll show you in the next picture. Sensation, though, was normal, so no clear involvement of loss of sensation or paresthesia or anesthesia, and also coordination. So cerebellar signs is what we normally say as a general term for coordination. So how quickly people can move, how precisely movements occur. Her gait was slow and she was not able to walk on her heels, meaning that the muscles that lift her toes up, or what we call dorsiflexion, were weak. And so this picture is basically with her looking as far to the side as she can. So you can see very, very little movement. You should be able to see a majority of the white of the eyes there. And so you can see very limited. So what does that do? Well, what that can do is make your two eyes not be able to line up correctly. So you may have heard the term diplopia or double vision. So if the two images from each eye don't overlap in the brain, or they vary how they overlap, then sometimes you'll see 1.5, sometimes you'll see 2 separate. And that's a pretty significant characteristic of disorders that affect that final connection between the nervous system and muscle, the neuromuscular junction. And so this clue made us suspect it was a neuromuscular junction disorder. So there are a couple clinical tests that we can do to assess how well NMJ transmission is working. And so in the normal healthy state, you probably have heard of the safety factor. The safety factor for signal from the nerve and activation of the muscle to trigger a muscle fiber action potential is redundant. And it means the signal's a lot louder than it needs to be. And that redundancy is what you probably have heard of the term safety factor. That's what that is referring to. So these are a couple tests that we can do to see if there's a problem of the neuromuscular junction. And I'm definitely making this case much more simple than it was. And when we get to the end of the story, you'll see what I'm talking about. But through a series of visits, we did determine it looked like she had a neuromuscular junction problem, a transmission defect. And so two tests that I'll show you very briefly is repetitive nerve stimulation. So that's where we stimulate the nerve and measure the response of the muscle electrically. And then also single fiber is where you measure individual muscle fiber potentials and see how consistent and how reliable those potentials occur, or action potentials of the muscle fiber occur. So just a high-level overview of the safety factor. So if the in-play potential reaches this threshold, it's kind of small, hard to see on that screen, but you have a small faint dotted line here. When the in-play potential or in-play response is reaching a certain threshold, action potential occurs. So what is the in-play response? It's basically packets of acetylcholine molecules that are released. They traverse the synapse and they activate ligand-gated channels, which create this influx of sodium. So you get an in-play response. If that small influx is big enough to reach threshold, then the voltage-gated sodium channels open, and then you get action potential generation of muscle fiber. That in-play response and that threshold, there's a lot of redundancy there in the healthy state. So that should really never fail under normal physiological conditions. And so when we measure neuromuscular junction transmission in patients, we can use electromyography. Normally we use a little bit of a more sophisticated version of it where we kind of zone in instead of a whole motor unit. We're looking at the muscle fibers connected to the same motor unit, so what we call single fiber electromyography. And you see every time this neuron fires, you get a motor unit. So this is all those muscle fibers that are controlled by that motor neuron. They fire in synchrony. And so when the person's trying to contract, it fires. Every time it fires, it should look the same because it's all or none potential of the motor unit. And then if you zoom in on that motor unit and look at the individual muscle fibers, each potential should look the same, should be stable. So the distance, the latency between those peaks is stable. The number of peaks is stable. And this is what we would call our term normal. In the abnormal condition, every time a motor neuron signals, not all the muscle fibers hear it like they should. So sometimes they activate, sometimes they don't. So you have intermittent neuromuscular junction failure, a transmission defect, and so this shape changes, and that's because all those muscle fibers that should be acting together, they're not all acting together in synchrony, so sometimes they're failing. When we zoom in on this whole motor unit and just look at individual components for muscle fibers, you can see the spikes then sometimes vary, sometimes don't occur, because action potentials of the muscle fiber are all or none, which you probably already learned. So they're either there or not. It's not like the action potentials are changing shape. The motor unit changes shape because it's lots of action potentials. But when you're down at the action potential level, and we do this in ways that we won't dive into, but basically it's a selective electrode, you can see it's either there or not, and the timing changes. So this is the most sensitive way for us to tell if there's a neuromuscular junction defect. I do these tests in patients most weeks for a range of disorders, including cases such as the one I just presented. So what does this look like on a video? So this is, I think this is actually my muscle, but this is basically a fancy electrode in a muscle while you're contracting. No stimulation, just activating. And it's 3 spikes that are happening in kind of at the same time because they're all connected to the same motor neuron. They're all within the same motor unit. So those 3 spikes represent individual synapses and muscle fiber action potentials. And so they should always occur together, okay? Now, what happens if we have abnormal transmission? This is, in this scenario, we're gonna have 1 motor unit with 2 spikes, so we have 2 muscle fibers. So depending on where the electrode is, you may see 1 spike of a motor unit, you may see 2, you may see 3, you may see 10. In this case, we see 2. So this is under voluntary control, not me, somebody that actually has a neuromuscular junction problem here, and you'll see it looks a lot different. Maybe. So you see how, you see that variability, and occasionally it's very transient, there's a failure there, and then we quantify it. So the red ones you can see are getting more spread apart. Sometimes it's superimposed, so it's hard to see. Sometimes they don't fire. And so this is a single motor unit. We're kind of triggering the screen, so we're taking snapshots of this every time it fires. And so that variability is increased because the in-plate response is not reaching threshold at the same rate, so the slope is not steep enough, and so then you have shifting of this time scale, and that's what we call jitter, and the occasional failure is what we call blocking. Any questions about that? Just stop me if you have questions. And this is the repetitive nerve stimulation that I mentioned that we performed in this patient. So normally repetitive nerve stimulation, we do at very low frequencies because as you go higher, you don't see as much defect. This patient actually had a little bit different pattern, and it's what clued us into something was different about this patient. One, most of the time when we see NMJ transmission defect, it's a disease called myasthenia gravis. That can affect a range of ages, and it's autoimmune, it's very treatable. But the pattern is normally you see more decrement at 3 hertz and less decrement or loss of the CMAP. And let me point out what decrement is. So see how this is really pretty flat? It's a little bit of decrement, but pretty flat. It should be almost totally flat there. We see 4.7% loss there, but it's relatively stable. Greater than 10% is abnormal. But then as you increase the rate of stimulation, we get more and more decrement. Normally, myasthenia gravis is the opposite. So this clued us into something was different about this patient. And so we subsequently did, and this has now been a long time ago, genetic testing and found, so she was the second person in the world, and the way she presented was a little bit different, to have a problem of the voltage-gated sodium channel. So basically this channel wasn't opening and closing like it was supposed to. So the end-plate response was fine, but the response to the end-plate response was altered. So then basically the muscle fiber became inexcitable, and that caused weakness, that jitter and blocking that we noted. So that was a scenario where we had loss of excitability, a loss of excitation at the NMJ, a loss of safety factor. And this was a genetic problem, but there actually were drugs that could help modulate the excitability that we were able to start her on once we figured out where the physiological deficit was. Okay, so that's loss of safety factor. I don't know if anybody, most people probably are maybe heard of Goldilocks and the Three Bears. So I like to use the term Goldilocks a lot because you want things just right. You don't want too much or too little. So every time the nerve signals, you should get one response from the muscle. So it's a one-to-one relationship. If you have an NMJ transmission deficit, sometimes that one-to-one becomes lower. So sometimes you get a response, sometimes you don't. What happens if you get more responses to each nerve input? So have people heard of the Tennessee fainting goats? If not, YouTube it. I was going to put it into my slides, but then I was too lazy. But they're really funny videos. You should check it out. So Tennessee fainting goats, if you watch the video, there's this farmer that goes around and like startles the goats by clapping behind them, and they basically become stiff and fall over. They're actually not fainting. What they're doing is they're becoming stiff because the muscle is activating too much. So losing that one-to-one to the other direction. So this also happens in patients. So as a neuromuscular specialist, one of my main areas of focus is a disorder called myotonia. Has anybody ever heard of myotonia or myotonic muscular dystrophy? It's actually the most common muscular dystrophy in adults, and it's one of the more common diseases we see in the clinic. Still very rare, and we're doing some early phase treatment studies to go at the genetic mechanism of myotonia. But I want to talk about a little bit about myotonia and some ways that we've been trying to treat this. So this patient is closing her fist as strong as she can for 5 seconds, and then we're asking her to open her hand as fast as possible. So imagine if you go to shake somebody's hand or you go to pick up a drink, try to do this, and your hand just locks and doesn't open. Or you're driving down the road and your eyelids close and you can't open. So, it's a very significant problem to have. So myotonia basically is the muscles just continue to fire and don't relax like they're supposed to. So it is a muscle fiber sarcolemma deficit where instead of being able to modulate the excitability gets kind of out of control. So myotonia can be looked at electrically, clinically, and mechanically. So we'll let that play through. This is EMG with no contraction. This should be quiet. This patient's muscles just going crazy without any activation from the nervous system. So that repetitive firing is what we call myotonic discharges. Same thing you can do mechanically or clinically. So here we're going to tap on the muscle here, and you'll see even mechanical stimulation causes the muscle to contract because their mechanical stimuli can activate muscle fiber action potential. So, and that can repetitively occur. And then these two tracings are actually a myotonic dystrophy mouse that we stimulate the nerve, and you can see in a normal mouse, it should relax and go down to baseline. Here you can see it doesn't relax like it's supposed to. So this is just basically a force measurement or a torque measurement. This is that same tracing that I showed you, the different frequencies. So this is a tetanic stimulation at 150 hertz, maximally stimulating the muscle, and then stopping the stimulation. It should drop back to 0, but in the myotonic scenario, it maintains. So we have looked at a few different types of treatments, and so a couple clinical studies where we looked at a treatment in paramyotonia congenita. That's a disease of the sodium channel, voltage-gated sodium channel. We also looked at it in myotonia congenita. That's the fainting goat gene that's affected. And I just want to show a couple videos. So you saw the patient before. So this is a patient with myotonia. Let's see if this will play. Maybe it'll just do one at a time. I don't know. She's going to close her eyes really tightly for 5 seconds and then try to open as quickly as possible. For the sake of time, I'm not going to wait. So we're going to have her close her eyes again here. She's still trying to open her eyes from the previous video. Closing her eyes for 5 seconds and then opening, and you see it's really pretty much instantaneous. And so how do we do that? Well, what we did is we looked at basically a mechanism of what patients with myotonia notice is the more they use their muscle, the less stiffness they have. So they have what's called a warm-up phenomenon. And so, well, we were like, what the heck's causing the warm-up phenomenon? So we had a mouse model of myotonia congenita, and we looked at the physiological like channel activations that might be driving that. And we saw that it was a late inactivation of the sodium channel that seemed to be driving myotonia. So almost the opposite of what that patient of the NMJ transmission deficit had. They have too much like recurrent activation. And there actually was a drug that was FDA approved for another indication, actually for chest pain in people with chronic vascular heart disease, called ranolazine, that we knew acted through this mechanism. So we reached out to the drug company and wrote a grant and said, will you fund this? And then we ended up doing 3 different clinical trials, and now this is used in patients with various types of myotonia. So pretty neat full circle from basic science, first learning from the patient, like what do they notice, taking that concept of warm-up, trying to understand it, and then basically trying to induce it with a drug. And now this is pretty commonly used. We're also looking at genetic-based treatment, so not just, that's a symptom treatment. We're also trying to address the genetic mechanism of disease. So this is a tracing that I showed you earlier, but we're also doing a genetic treatment in these mice through knockdown. So they have a repeat in a gene called DMPK. And so they have a trinucleotide repeat expansion, and when that gets expanded, it gets turned into RNA in cells. And then those foci of RNA alter the way that different genes are spliced, because these foci of RNA cause RNA binding protein sequestration. And so what we're trying to do is knock down these repeats that accumulate in cells, and you can see that we're seemingly getting some early signal. And there's a number of clinical trials that are doing mechanisms. We have a new early phase study that's getting ready to start patients. So we kind of do things from both the preclinical side and the clinical side. So I don't want to run out of time and not give you time to ask questions. I do have the last case-based thing that I could talk about. What do you prefer, questions or keep going? Too shy to ask questions? It's okay if you are. Questions? No volunteers? Okay. Spinal muscular atrophy, I mentioned this. So the most common genetic form of death in infants, roughly about 1 in 10,000 live births. So there's a range of clinical severity, and I'll talk to you a little bit of why that is. So it's genetically, it's a cool disease, not to have, but to study. But patients present with generalized weakness. On average, untreated babies with SMA, they normally have symptoms by about a couple weeks, a few weeks. They normally die by about 10 months of age on average. And 95% of babies die before their second birthday without treatment. So the genetic basis is very interesting. So the causative gene is a gene called survival motor neuron 1 gene. So this is an autosomal recessive disease. So you have to inherit a mutated version of the gene from both parents. There are usually large deletions of this gene that cause SMA, and you have to have 2 of those copies. Occasionally, you'll have a small missense mutation. Occasionally, you can have a de novo mutation. Has anybody heard of de novo mutation? That's where a mutation wasn't inherited, it just happened. So about 1% of SMA, one of the copies is de novo mutated. But most of the time, 95% of cases is homozygous large deletion of the SMN1 gene. Now, humans are unique in regards to this gene because we have SMN1, but we also have SMN2. We're the only vertebrate species that have 2 copies of the SMN gene. And I kind of simplify this, that SMN1 is like the fully efficient gene. SMN2 is the wimpy backup version. So it's a highly unstable region of the genome. And so these have been duplicated, but there's a nucleotide change between SMN1 and SMN2 that makes SMN2 not very good at producing the full-length protein, which we call survival motor neuron protein or SMN protein. So all the transcripts essentially from SMN1 are full-length. And so they're stable, they oligomerize like they're supposed to, they do the job that they're supposed to. SMN2, though, can only produce about 10% of this full-length protein. So if you don't have an SMN gene, across all species that has ever been studied in, it's incompatible with life. Like you're not born, life ends at the multi-cell stage. So if you knock out a mouse's SMN gene, they're never born. Same thing with various other species. So without the SMN2, you don't get SMA. So that little bit of protein that's produced by this wimpy backup version gene allows patients to live, but it's not sufficient for the motor neurons to function like they're supposed to. So if there's never been a report of a human that didn't have either gene, so mutation or loss of both SMN1 and SMN2, because you need some SMN protein. So this is interesting because this protein is expressed everywhere. But when it's low, it seems to be relatively selective impairment of the motor neurons. And we actually still don't know exactly why that is. So this is ubiquitous, meaning it's expressed everywhere. Levels do seem to vary between different tissues and during development and aging, but we don't know the link of like when it's below this level, this low levels just from SMN2, why the motor neurons die. So you can see 3 pictures here. I don't know if it played while I was talking. So a younger baby, which you saw earlier. So very prominent weakness. You'll see like head lag there because of neck weakness and proximal weakness. You can see very bright and alert, but really, really weak and hypotonic. And then you'll see a patient kind of in the middle that actually is able to run, not perfectly, but able to run. And then you can see this child has kind of braces on and can kind of ambulate with a lot of assistance. And so this is kind of the range of SMA people typically call SMA type 1 is the early infant onset. 2, they're never able to sit up on their own. And 3, patients are able to walk. Type 1 is by far the most common. Those infants are never able to sit up on their own. They generally have 2 copies of SMN2. And so that's what drives this range of phenotypic severity. The SMN2 genes vary across the population. Some people have 2 copies, some people have 1 copy. Occasionally you have no copy, but you wouldn't have SMA if you didn't have any SMN2. And you can have as many as 5, 8 copies of SMN2. So you get a little bit from more and more copies, so then you can kind of reduce the phenotype. So basically we have biological evidence that increasing the protein should modulate the phenotype. And so what we did is, I'm not going to tell you the whole story, but it was a story that took a little less than 10 years. So this mouse model, so the gene was discovered in 1995. The first mouse models were developed in 2000, but the one that we used mostly was in 2005. That was developed by the person that kind of mentored me as a scientist. And then we started working on gene therapies around 2009, 2010. The first really successful publication of gene therapy in SMA was in mice in 2010. We developed a large animal model of SMA, and then we moved to a clinical trial in SMA patients around 2013, 2014. And so there were times where I was part of all 3 of these stages of development. So mouse studies, a large animal, and also first in human. And so this have people heard of the different phases of clinical trials? So phase 1 is purely safety, so to make sure you don't hurt people. That's typically in healthy volunteers. Phase 2 is early efficacy intervention. Phase 3 is really the definitive, does this work? And then phase 4 is like after it's available, kind of monitoring it in patients in real world. So phase 1 typically is in healthy individuals, but our approach was using a gene therapy. So because we're modifying people genetically, you really can't do that in healthy control. So met with the FDA about moving this forward to patients. And basically what we're doing is we're taking a virus, a viral vector. Adeno-associated virus is the typical vector that people use. It's not pathogenic. It doesn't insert as cargo into the genome, so it's what people call a non-integrating vector. And we packaged a gene that was basically to replace the SMN1 gene. So this fully functional SMN gene. And so then this is delivered in the bloodstream, so venous delivery, in the case of these babies, over 1 trillion viruses. And basically it delivers the cargo across the whole body. Now, because it's non-integrating vector, cells that are not post-mitotic, that are still dividing, that's not going into the genome. So once they divide, you lose it. But the value, or the cool thing here, is the motor neurons are post-mitotic, so they don't divide and you don't deliver, or you don't lose that delivered cargo. And so we did this first in human study. And the natural progression of SMA, you can, we have shown that SMA patients that are untreated never reach above a 40 on the scale that's called the CHOP intense score. You don't need to really memorize that, but they should never get above this score. So phase 1, you normally start low dose and go to higher dose. So the first 3 infants got low dose. They still did well, but they didn't reach those functional markers like the other babies did. And you could see that these mostly all got above that level. Interestingly, there was a clear age and time dependent. So time was motor neuron. You go in earlier, you have more of effect. And so unfortunately, this later treatment participant didn't have as much of effect. Still had some effect, but really didn't have that dramatic improvement. And it was interesting because I had done all these studies, in other stages of research development. And if anything, I think the human studies showed more of a response. You always hear that, moving things from the lab to the clinic, things don't translate and things like that. But in fact, I think in this scenario, we actually saw a bigger effect than we did preclinically in other models. And so we're still learning about the biology of SMA. We did this first in human study in 15 infants. Now this is used all across the world. We treated the first baby here in Missouri after I moved here a little over a year ago. So this is being used everywhere and changing the landscape. And there are actually now multiple different types of treatments, which I've also been involved in. And we have the next generation of treatments for SMA that we're actively enrolling in a clinical trial at NextGen currently. I'm going to show just a video. So Evelyn, she was also number 6. This is her at 19 months. Her sibling died in 13 months, was never able to sit up, right, walk, or do all talk and eat by mouth and all those things. And so she was treated about 6 weeks of age. And you see that she clearly still has weakness, but she should never be able to sit upright on her own. So really, as a physician, really amazing thing to be part of. So now we have 3 different treatments. And so I told you about this Wimpy backup version gene. There also are ways to make that gene work better. And so the 2 other treatments actually make SMN2 act more like SMN1. So they increase the efficiency of full-length transcripts and full-length protein of the SMN protein. And all 3 of those are in clinical implementation and use now, which, you go from this universally lethal disease now to a disease that we have 3 treatments. And it's really an amazing thing. So with that, I think I have just a little bit of time if you have questions. Hopefully that was valuable, high level, but kind of what a neuromuscular person does clinically and what I do in the lab. Questions about anything? Not curious at all? Well, we'll work on that. Great, thank you so much.
Thank you.
Are you reading me?
Yeah.
Oh, please.
I have 2 tests to take.
If anyone's didn't get the sheet, I have it right here.
Well, I have. You have a test today too, sir. I don't want to hear it. Oh.
Thank you. Oh, it's right here.
Yes. Because I love you.
Facebook that you won a grant.
Oh, yeah, we
She sent
just,
it to me last night and she was like, this is so cool, you should like look into it because I want to go into neuroscience.
oh, cool, awesome.
And she was like, I was like, so.
Wait, how do I do it?
Actually lecturing in my class.
Tomorrow.
today.
Yeah, so that, our interest of firing rate of motor neurons, it says it has